Dr. Kevin Haas, thank you for joining us, is a life sciences and technology leader with over 15 years of experience in genomics, bioinformatics, and digital health. He most recently served as CTO at Myriad Genetics, leading development of precision medicine platforms and data-driven approaches to genetic testing. He has held leadership roles across engineering and bioinformatics and helped scale innovative diagnostic technologies. He holds a PhD in chemical engineering from the University of California at Berkeley. So please welcome Kevin. I'll go in order. So Dr. Sowmi Utiramerur, VP, Head of Translational Research Oncology and Genomic Medicine at Roche. With over 15 years of experience in clinical genomics, computational biology, and translational research, Dr. Sowmi is a research leader across academic and corporate settings. At Roche, he leads research programs spanning clinical genomics, histopathology, immunohistochemistry, and computational biology, focused on developing diagnostic and prognostic assays across oncology, inherited diseases, women's health, and infectious diseases. His work in precision medicine, including LDT and IVD assay development and biomarker discovery, has earned multiple innovation awards and patents. So please welcome Dr. Sowmi. Dr. Pauline Fujita is Global Head of Analytics and Workflows at the Computational Sciences Center of Excellence, CSCOE for short, at Genentech Roche, where she leads teams managing high-dimensional data platforms, workflows, and analytical tools. A bioinformatics leader with a track record of developing clinical-grade software, she leads the development and adoption of scientific software, data products, and machine learning-driven solutions that support research at scale, enabling scientists to translate large-scale data to meaningful insights and accelerate discovery. So please welcome Dr. Pauline. Dr. Laure Frésard is, I hope I'm pronouncing that right, is Senior Director of Translational Genetics at GeneDx. She brings over 15 years of experience in molecular biology with a focus on leveraging large-scale data sets to understand clinical phenotypes. At GeneDx, she leads translational research aimed at improving diagnostic yield from whole genome sequencing data and advancing rare disease diagnosis. She holds a PhD in molecular genomics from Université Paul Sabatier Toulouse III and completed postdoctoral research at Stanford on integrating expression and splicing data with genomic sequencing for rare disease diagnosis. So please welcome Dr. Laure, Frésard. So it's great to have all of you. Thank you again for joining us. So we have some seeding questions, and maybe we have about 20 minutes and 5 minutes Q&A. So we can just keep it very open. So we just have, feel free to completely ignore these questions if they're not very good and answer a different question instead. Maybe I might deviate a bit too, because we've been, so we'll all talk about AI, I guess. Sort of almost a given now that we always talk about AI. I wanted to focus on a couple of different things that we've been looking at Strand and get sort of an opinion and maybe a discussion. One is the use of AI in software. So just for speeding up software development, let's say, right? So just as a tool, right? So for instance, Cloud Code or the like. The other perhaps parallel use case for AI could be, and we read the paper from GeneDx sort of, I don't know how much of it is AI-based, but certainly you are speeding up rare disease variant interpretation as well. So it might be interesting to think about it from a diagnostic perspective, or how you might be using it at Genentech or at Roche DX, but potentially not so much from a software development perspective, but more from a fundamental biology perspective. So I was wondering if, Kevin, if you could kick us off and just comment on how, maybe from a software development perspective, has AI changed the amount of time that the average developer spends in molecular biology and bioinformatics in developing a piece of code, and how you see that?
Yeah, no, great question. I feel bad because I haven't had the chance myself. I did all my bioinformatics before AI was invented, at least the most recent cogen pieces. I do remember how long it took to be able to get to a functional pipeline. And I think we showed Sam's cat took a fair amount of time to get 17 minutes or something like that. But it was months if you were new to the stack to figure out even just the most basic things of like, how do I even publish this? How do I write a WDL pipeline? That all looks really weird. Or if I want to make a Nextflow one instead, what are all these parameters I need to do for BWA? And I think the amazing democratization is that, especially in bioinformatics now, you can basically vibe code your way to a pretty solid pipeline in like an hour. It's just because you can get embedded within some of the cloud tools like Healthomics, for instance, on AWS, that has plugins that have been optimized for cloud code and for Amazon Zero IDE. So you can really just prompt it like, I'm trying to do a whole genome RNA expression assay, and I want to find out, log 2 deviations between this just describe, and then it will publish it as a compiled pipeline ready to rock and roll. And so I think the innovation in bioinformatics is that it's really democratized that in a big way. It used to be you had this huge wall between the people that could do the wet chemistry, the people that could do the dry lab and analytics, the people who could do the software. It was like a 4-person handoff type of thing. And that was the only way you could do it because each domain was too specialized to do anything else. And not to say it's like completely turnkey. We talked about some of the infrastructure you have to do, but I think you're seeing there that at least your first forays can be much more attainable to, and now someone can be much more not a polyglot, but they can span the whole nexus of different capabilities within the bioinformatics space.
Thanks, Kevin. Can someone else maybe, I don't know, Sowmi, could you please comment on in a science or maybe even along the same lines? I mean, if maybe we just pass the mic along and people can share.
Yeah, I guess I'm not the best person to talk about how AI is increasing productivity in software development or bioinformatics code development, but I can speak in terms of fundamental understanding of our biology and the inference, right? When it comes, we are all mostly in the diagnostic space, right? If you take a particular example of diagnosing a disease, right? All of the basic markers, right? If you have an elevated level of a certain biomarker or a molecule, you have these diseases. So the simple diagnostic markers have all been solved for. Now we are in the space of complex diseases where there are multitudes of biomarkers that interact with each other, right? So we generate so much of data in order to be able to better understand the interactions between these various molecular biomarkers in order to see what is the accurate diagnosis or what is the prognosis for these various diseases. But as humans, we are not able to interpret this data that we generate right now. And that's where the complexity is preventing us from being able to do either better diagnosis or better prognosis for any number of neurodegenerative diseases, right? If you take simple things like MS or like depression, for example, right? So many people have bipolar diseases, other depressions. We have only a pathological diagnosis for them. Like there's no molecular diagnosis for any of these diseases primarily because they are complex, right? We can measure the number of markers, but we cannot correlate them to what is the causal thing, right? Because there's complexity there. That's where we are starting to use AI to be able to better understand the causal correlations between the various markers. You can generate as much data as you want, but as humans, we are not in a position to be able to make inferences from this data as of now, right? And that's not going to change. And that's primarily where AI is coming in for us to be able to make better inferences, better understanding of the biology, fundamental biology. And that's where we are using exploratory in diagnostic use.
Thanks, Dr. Sowmi. And Dr. Pauline, if you could comment, I don't know, please feel free to chime in, but specifically maybe AI as a time saver versus Genentech has, I think Aviv Regev's group has released several pioneering deep learning models in biology, right? So that's a completely separate realm of you're trying to predict cell type annotations as an example, or you're trying to do something much more fundamental, right? So could you please comment on, is that a useful distinction as sort of one, just using AI like Claude to, hey, what's a GWAS pipeline look like? What are my QC metrics, right? saves a lot of time, as Kevin was mentioning as well, versus something much more fundamental, like actual, like a biology foundational model, for example?
Yeah. So it's interesting that the team that I lead, our stakeholder base spans their AI literacy from I'm trying some stuff out and I'm vibe coding and some of it's slop and I can't tell, to I'm making new things that are new models. I have a better informed technical opinion on what I'm looking for in terms of the computational support. And the challenge with the team, I think it's a lot of what has already been said around the efficiencies that we're gaining, around the democratization of who can access those. I think, so I came up around the same time in terms of bioinformatics before all of the AI stuff. And so there's a lot of, it's a really interesting time in industry because I think a lot of the cachet of bioinformaticians was around your ability to flex for your knowledge of syntax and configs. And if you'd written a pipeline in a particular language, it's then bound to Nextflow or bound to WDL or bound to CWL. And that would be the reason that there would be a silo around that methodology. And it would be very expensive to rewrite that in something, a different implementation. And the cost of implementation has gotten to the point where a wet lab scientist will come to us and say, I made a thing, seems good enough, like here, you take it. And so that and my team has really been trying to figure out what is it to reimagine that activity in this new AI native context. What's the role that we play now, where some of the things that we used to be, where it was important for us to be involved because we could provide that service of translating a human thought into a bioinformatics pipeline or some syntax. What are we doing now? And what I've been telling them is that I really think it's kind of like we run a tool shop and we really have to be the experts in which tools are good, which saws are rusty and you shouldn't use those, and also knowing what good looks like so that we can partner with people that are coming to us that have made a thing, because everything's been democratized to some extent, but the ability to tell what's good, what's reproducible, what's good science, what are decisions that we feel good about, to put some guardrails in place in terms of how we support that. And equivalently, I think for folks on the ML side, a lot of times they come in the door with this really deep expertise in the AI ML space, but they're not as good, they're not as well informed in their consumption of omics data, for example, in terms of I could hand them genomes that are run on a really old pipeline where I can tell you for sure that the metrics off of that, the data's not what you want. And we have to make smart investment decisions around if we're going to blow a bunch of money on tokens and GPUs and all this stuff to run the ML, but that the data I'm going to put in is from a pipeline that's 10 years old and I know there's regions of the genome that can't catch. They don't know to ask for some of that stuff. And so we kind of sit in this translational layer between science and engineering and ML. So it's a really interesting time.
Yeah, the domain piece is the differentiator. Dr. Laure, I think your domain is slightly different, perhaps, in terms of rare disease diagnosis, if I'm not mistaken. I was curious if you could comment on either the use of AI or even more traditional, decision engines to speed up diagnostic, tools. And
absolutely, yeah. No, I think in the context of rare disease, where AI is helping us and can help us a lot more is in helping us prioritize and structure data. So prioritization is reduce the search space. So for all of our scientists is how do we go from pool of candidates to the most likely candidates to look at and focus their time on those most important. So we have tools at GeneDx, like called Mimi Ranker or GEM, that are specialized in those, on like how do you predict the most plausible diagnostic variants so that scientists have less variant to review. I think it helps us also elevate the right evidence. I think it goes back to how do we make sense of all the data while elevating and ranking that evidence in a purposeful way is another way AI can help us in the context of rare disease. For example, having a good understanding of gene disease association for rare disease is key, but it can take time at the time of interpretation to make sense. Well, is my variant in a gene that makes sense for the phenotype I have? Not always obvious. And that's an area where AI is helping us because we have models that are combining external data like oming or publications, combining them with our own data set and our own structured data sets to make sense of it and triage. So this tool is called multi-score, and it helps elevate those most plausible way. And I think a last area where it really helps us is how do we extract structured data out of everything that is available? And it's more and more important. Like publications are keeping going on. There is clinical notes. How do we parse them in a way that is systematic and that we can trust and rely upon for our internal analysis? So I think all those fields are how do we assist clinical experts in making the right call and elevating the right case for them.
Yeah, thank you so much. It's very interesting that there's just so many different applications here in this panel. It will be remiss of me not to use some of our pre-prepared questions. So if you don't mind, I will direct one each of the prepared questions, right? So Kevin, if you please, so I'm just going to read out and please feel free to respond to whichever part of this is most appropriate. What are the key challenges in validating and deploying AI models in diagnostics in the context of FDA's evolving guidance on AI ML-based software as a medical device? That's interesting, actually. How can diagnostics organizations effectively integrate AI tools into their workflows while ensuring scalability?
Yep. No, it's a great question. I think the interesting thing is that the FDA does have actual guidance about how you could deploy an AI tool in medicine. So it's not like it actually follows a lot of consistent principles. It's what's a risk-based assessment? How do you do your standards of validation? What is reproducibility? How do you do rare event and adverse event outcome tracking? It's like a lot of standard QMS type or quality management system type theory. I think the biggest challenges and differences is first on the risk-based side, which is that this, if it goes wrong, can be bad. And even a little bit of a myriad story is we did AI applications in our production workflow, but most of those were in the billing space. So AI bots fighting with insurance companies to get reimbursement, where the worst case scenario there is it's just money. But obviously when you're trying to do in medical applications, I think the thing that's the biggest difference though is the non-determinism of the modern AI models.
Yes.
Because it's not like people haven't been doing AI. I mean, deep variant being used in production pipelines has been around for 8-9 years or something like that. So it's not like people haven't done black box AI models that were low explainability. I mean, all the image analysis models in digital pathology or digital radiology, they all have a certain element of that. But the fact that they're deterministic makes it a lot easier from a regulatory perspective because they can be reproducible, they can be studied, the blast radius is sort of bounded. So I think it does give some just sort of technical best practice, which is even those large foundational models will always be the most advanced. They'll probably, any customization you do in 8 months, a foundational model is going to beat it anyway. So it's almost not worth making special bespoke models unless you have special bespoke data, like a AI drug discovery company. But the good thing is if you get really small models that are just distilled, you can get much closer to reproducibility and determinism, and then you get a much less of a variability in the outcome. And I think that would lend itself to being much more easily either edge deployed just from computational use or much more like bounded in the study of its performance. And but I would actually be interested almost just for both the panel and the open area. I don't know if anyone's actually launched a product yet using the FDA's guidance of how to do a software as a medical device using AI.
Using AI.
Yeah, and maybe they're in the clinical decision support case. There's been a couple of examples, but I was thinking right before the panel, I didn't actually know of one yet deployed.
Would the pathology companies be the first in this?
I guess those wouldn't be LLM based. Those would be,
wouldn't be
they'd be
LLM.
imaging.
Yeah,
the imaging side, like convolutional neural nets, obviously tons, but
Right,
I don't
tons.
know of any LLM based or that generative AI generation.
Yeah, I mean, wouldn't a challenge be what you mentioned, which is that you can't predict the output with 100% certainty?
Yeah. If you get really small reducible models and you get really tight control, you can dial down the temperature to them to 0 and you can get them to be deterministic.
Right.
But you just can't be downloading, Claude Opus and just seeing whatever happens. It's got to be a really tightly controlled environment.
And how do you measure the accuracy of something that keeps changing its output? And what are the fixed points in the output that against which you can measure the accuracy? I guess those are all questions.
Yeah, constantly learning model sounds scary to have that in production.
In a medical device,
yeah, you're
sounds...
like resubmitting your 10K hourly. That just seems a little bit strange, but yeah. But also at the same point, though, it's definitely where we're going. Like, I think it's completely naive to think in 5 years you're not going to have the majority of diagnoses in the medical field being done by AI too. So it's both incumbent and inherent that that's where we're going to go. At the same point, with the current technology, making it so it's actually all above board and done with the proper safeguards is, I think, still a challenge.
And I have a great follow-up for Dr. Sowmi here, which is, how would you validate and refine these AI findings in the R&D space? What resources will it take to validate, all of these findings in the R&D before you can translate them to, exactly this very thematic, I think. So, to clinical workflows, and if your take on this, please.
Yeah, I feel that we are far, far away from being able to do validation and putting it into a clinical diagnostic setting, right? Maybe on the clinical decision support side, yes, we are much closer, right? On the digital pathology side, yes, we are much closer. But if we are generally talking about, like, making a diagnostic from a multimodal or a multi-omic biomarker, that we are far away from, right? The reasons why I'm saying that is because, right, we are first far away from first understanding the fundamental biology, as I mentioned before, right? First, we need to be able to do that. For that, we need lots more data that we are from what we are currently generating. I give you a very specific example. Right? Again, Genentech and us, and I'm sure hundreds of pharma companies out there are all doing perturb-seq and single-cell sequencing data in order to be able to better understand, right, going from a CRISPR knockout of a single gene to what the phenotype change of a cell is going to be. There are hundreds of tissue types, cell types, thousands, 10, 20,000 genes. So you take the combination, the amount of data that you need to generate just to understand a single gene knockout correlation with the phenotype. Now, we all know in real biology, cells don't work in isolation. Right? If you want to understand how a tumor cell behaves, if you want to design what the target is in order to be able to kill a tumor cell, for example, it lives in a complex tumor microenvironment, and all of the cell interactions underplays what the transcriptomic changes are, right? And also, we know that somatic changes and genetic modifications have a correlated effect in all the overall phenotype. It's not just the somatic mutations are independent and having a phenotypic change, right? So how do you understand the relationship between the somatic mutations and the underlying genetics that the patient had in order to be able to understand that? And then top of that, you have haplotype insufficiency, multiple gene correlations. So you can start to understand the amount of data that you have to generate in order to fine-tune or train a model to understand the biology before we we start applying this in any clinical diagnostic setting out for target discovery, right? So yes, we will get there, right? We are starting to generate the data at that scale, right? But I would say, right, the faster way we could get there is to say, okay, from hypothesis driven, again, people don't hate it because you're going back to where we started from, like, hypothesis driven data generation and then trying to understand the biology from that. But I would say we have to find a middle ground. That is that you start with a hypothesis, generate multimodal data, multi-omics data, use that to specific, not just go straight to a foundation model type model, but to be able to have a very specific model for the problem that you're trying to address, right? Whether it's in oncology or neurodegenerative diseases or rare diseases for that matter, right? And then develop that model with the specific data sets that you want to generate to answer the hypothesis. That's where the models can be used to do better diagnosis or prognosis, and that's where we can potentially lead to a clinical diagnostic setting, I feel. Yeah, that probably will help us get there faster than just saying, okay, hey, we are going to develop a generic foundation model that is going to help answer a diagnosis, right?
Solve biology. Dr. Pauline, I have again, along similar lines, production-wise or even in R&D, what roles and tasks are being augmented by chatbots that are sort of more LLM-based, can read human grammar, understand in quotes, in large-scale data curation and working with real-world data? And what are some of the challenges associated with the auditability of GPT-assisted curation decisions? I apologize if that's a very specific question. I can make a more generic question out of that if you like.
No, I mean, we can work with that. And I think some of this has, I think, come up in conversation with your group as well in terms of the spaces that you operate in. I think it's like I was saying before, that it's really changed things that used to be intractable problems around things that are siloed, things that are too heterogeneous for us to solve or do things at scale, missingness in data, all of these things, our ability to really get to different levels of scale and complexity that are now accessible for compute. A lot of the stuff that, and I should caveat that with, my team sits in the preclinical space. We're not under a lot of the constraint that a lot of this room is experiencing in a way.
Yeah.
But I have had that experience in one role ago where I have some sense of like where some of that could intersect, and we're trying to also pay attention to it, because stuff that will go through a clinical context will then kind of come back around because there's an appetite to reuse that data or do something different with it.
Yeah.
I think in terms of where chatbots really come in, not just for curation, but in general, as we're interacting with scientists that don't necessarily have the same computational background, or where there's a burden, especially in the context of doing variant curation and things like that, where there's the scale at which I can ingest information as like a summarizing tool, as long as there's enough guardrail that I'm paying attention to, are the papers that it's looking at real, things like that. But there's a lot of gains that we're starting to see in that space and what that looks like. And there's even been papers done recently looking at contrasting the contents of the different biobanks, what the ethnicity profiles of those look like, what it looks like to bring those together, if the data model behind one and the other is different. Until fairly recently, that was like, well, we're just going to have to operate on them separately. But now, because the cost to do some of the implementation, to do data engineering and get to an interoperable state between different data assets, it's just that much more tractable now, that I think that a lot of that stuff is going to become a lot more accessible in a way that's even difficult to speak to, because it's changing so fast. Because it's interesting, I had somebody on my team that was on an extended leave of absence, and when they went out of office, everyone was gung-ho on Cursor, and by the time they came back, we'd moved on to a different tool, and he's like, Pauline, I feel like I just fell behind. I was only out for like a couple of months, but the field moved so quickly. So I really think there's a lot of promise there around some of the things that in this field have felt like this is the hill that we just, like, reanalysis is something that I think about a lot. In the previous role where we were building stuff to think about variant curation, that was a hard problem. Automated reanalysis, how would we ever do that? The engine to make that was just not possible. Now I could easily imagine how you might get to some of that, leveraging what AI could do for you, something that could scan all of the literature, tell me when relevant things have been,
With updated, so your VUS becomes a patho, ELP or whatever.
Yeah, exactly.
Yeah.
Sorry, I didn't mean to steal your question.
That's very interesting. Would you like to riff off of that, or I have a question for you instead?
I think through a question I can fall back on that.
I mean, I guess what level of human in the loop review is now a part of, I guess, playing on the auditability piece, perhaps riffing off of a different piece. Is it necessary now if you have AI-assisted variant annotation and interpretation now in the loop, and how much of human in the loop, therefore, do you see is super valuable, especially in a clinical setting now, which is completely, I guess, separate from our research setting potentially?
So yeah, I think that's a great question. I think human in the loop is still key. Like, humans are part of the process, but I think their scope shifts, and it's how AI is helping solve easy things, but how humans are involved in the high value stake or the high risk stake. And I think that's how I see the biggest shift. As I mentioned earlier, I think AI and machine learning are helping us be faster and be more efficient at getting to the answer. This is true in the clinical settings by prioritizing variant better and helping them. The level of risk is different. So at GeneDx, we have research and we have clinical settings. So we are developing things on the research side, and where we can try plenty of things and say, look, this is going to solve everything. And then we have to be down to earth on how do we implement those in a workflow where humans are helped and in the loop to understand, help solve the conflicting results, help validate those results, and help sign up the final report, because they are the one responsible for the output ultimately. And so I think those are key things in clinical settings. One, I would still say that there are some scenarios that we see in clinical settings where human can now be out of the loop because of AI. For the super obvious known knowns, this is a positive, like everything goes in that direction. We don't need to spend time, precious human time on those. Let's use AI to help us pinpoint to the case where they need to be focusing on to get what is the next evidence that we need to generate or to understand. Where do we need to go next to help solve all those negatives? And so I think human is not out of the loop, is key part of the process, but AI makes the human more efficient at what they're doing now.
Yeah, what we are seeing a lot as well is not clear that we can ever remove any humans, but definitely becoming more efficient in specific ways. I'd like to open it up to the audience for the next 5 minutes or so, and then we can call a close to the panel. Anyone want to go direct a question to the panel or to any specific panelist? Now is a good time. Christian. When are we going to have a digital twin?
As I mentioned, right? Digital twin is a very broad term, Christian. So what we are working on, CZI and others, right? Most of the people in this space we are primarily working on a virtual cell, right? Then we might go to a virtual organoid and then to a digital twin. So there's, as I mentioned, even in the virtual cell, there's lots of single cell models that we can leverage, but again, the first thing that we are exploring right now is the cell-cell interactions, right? Put a tumor cell and an NK cell in a vessel, and can a single cell model predict the transcriptomic changes in the tumor cell or in the binary model system, right? Or not, right? So that is the first step we are even exploring right now to get to a virtual cell that can predict some phenotypic changes, right? So then duplex to triplex and other complex systems, then we will get to an organoid. So how far away are we? I don't know, but we are just starting to scratch the surface, going from single cell to a duplex and to see the interactions there.
Thank you, Sowmi. Any other questions from anyone?
I was wondering whether you guys have in your organization that you have make an effort on the automation, on the workflow, more on the web lab basis. I know you guys are mostly talking about the data, but maybe the data is running out and you need more data to actually mine them and use the AI for.
I can do a quick response on that one. Actually, just kind of cool because we're in this facility. If you go downstairs, there's actually a pretty highly automated lab that does the NGS sample processing. And that actually kind of has nice windows and stuff. You can see it's a bunch of robotic arms on rails, and there's like a hole in the wall to pass things from pre-amplification to post-PCR amplification. And it really does automate probably 100 steps of wet lab processing, and there's several hundred robots that all have to kind of work in perfect harmony to make that possible, both from like low-level mechatronics to high-level lab orchestration and scheduling. So I think those labs are out there. I think we just do, 500,000 samples out of this facility, but especially when you're trying to do lab in the loop for AI drug discovery, you're talking about experiments that go into the millions a day type of a thing. It gets really, really quite impressive. But that does push the envelope on just like how good you are at orchestration, and also ends up being most of the challenges are what happens when things go wrong. So like 90% of the time it works perfect, and then the 10% of the time we got 400 things, something's going to break every day. And so a lot of it is like, how do you recover automatically from errors? And then you have to automate every single little step in the process. I think my favorite little robot downstairs is you get a box of tips from Tcan. They come with a lid on them. And so you got to take the lid off. You need a robot to take the lid off the tip box. That's like the de-litter. And every little thing, if you don't have a human in the loop, you got to kind of address every little piece of that, from waste treatment to then on out. But I think at scale, the AI drug discovery companies are putting some of these diagnostic labs to shame with just how many preps they do and how many experiments of perturb-seq they can run.
I saw his hand first. Sorry.
So if you interview someone and you are ready to hire someone, what question you will ask in your interview about the AI or about that person related to AI?
What's on the test?
It's interesting because we're at a time where the people's AI literacy is very bimodal, it really depends on the seniority of the role that you're hiring for and what you're looking for them to do. If it's a more senior role, you're looking for somebody that has a nose for what good looks like in the context of using AI and has some ability to flex in terms of what the vision and the intention should be in that space. If it's somebody a little bit more on the junior side, then they're more recently out of school and having come through a context where AI programs, like folks that are already in industry, would more likely to have taught themselves AI because it's been happening as they've been working. People coming fresh out of school or grad school would have had more direct exposure to formalized training. So it's a bit of both. And then I think we're having to reimagine what things like the coding interview even look like, but even just generically the interview itself, having a sense of, is this person using AI on the side as they're interacting with me? So some of it is just figuring that out, but also acknowledging that we do want to get to a state where everybody is using it and is not just conversant, but can use it really well. That's going to be a differentiator. And if you're not a person that's doing that, then you're somewhat getting left behind. So some of it is probing along. How are they thinking? Is AI just the way for them to go faster? Or have they been able to be creative with it in terms of being able to unlock things they wouldn't have been able to do or explore problem spaces in a particular way? So some of it is how they think, and then whether or not they've had the curiosity and drive, given where they are in their career, to have upskilled in that space. I don't know if that was too hand-wavy to answer your question. I saw you nodding out of the corner of my eye.
No, I couldn't agree more. I think interview-wise, the space have shifted very fast when you interview virtually. And to come to your point on like the real answers are really easy to find now and to get live. And so it's been more work, but good work into reimagining on how to ask the right question. And I want to relate to how do you think critically? How do you generate hypothesis and not absorb information while kind of thinking through how AI can help you build towards that as a tool to answer that question.
This question is for you. Two more questions. Two more questions.
It's a kind of follow-up on what we have been just talking. Is there any future for new graduate students in bioinformatics jobs?
What is the, may I rephrase that to what is the future of bioinformatics jobs, perhaps?
So I think that it's still important to have a technologist perspective on what it is we're implementing. And because I think, I have people even on my team that have self-trained to use AI, but that are trying to figure out what it is to not just use it to be lazy about the bottom 20% of tasks, but to have some discernment about how I'm using that as a force multiplier for what I'm doing, how I'm fitting it into my thinking, how it's improving the overall quality and volume of my work, not just the volume. And so I think for people coming out of a bioinformatics training, it's still about how they're exploring problems and how they're using it. But I would imagine, I mean, it's been a while since I've been in school, but I imagine there's a part of their training that's figuring out how that fits in and how they start to operate in an AI native context. And so I think a lot of the newer candidates that we're seeing come in the door having both done bioinformatics generically, but also used AI or built AI or done at least like the first 20% of being more of an ML hat wearing engineer in that space. And so whereas before it was sort of bioinformatics plus engineering experience, now it's bioinformatics plus ML engineering experience. Have you done stuff with models? What have you done? What does that look like? Because I think we still have to operate in the space where you do a lot of translation between a science question that you want to answer and the computational approaches you want to apply to it. And so there's a need to have people that have bioinformatics, people typically have sort of that hybrid thinking where there's been some exposure to the domain expertise of what the problem they're trying to solve and then how that fits in. And so I think it is going to be an area where what that role looks like is shifting quite rapidly. But I think domain expertise, I think, is going to be a place where it continues, value persists because we need that as we're consuming AI. And it is a place where there is a lot of people have been trained to kind of be discerning on how they find that information and get the right context for themselves if they've been smart about how they've been navigating their training. I don't know.
Last question, please. Yeah.
Online.
Online.
We were like
not quite energetic. So online questions, we'll have to address them at a separate time, perhaps. A lot of energy in the audience.
Okay, thank you. So going back to the fundamentals of biology, right, and the AI, we know that it's very complex to understand, like, and incorporating AI into it, going back to the single cell, trying to build a model, different kinds of data coming together, different modalities. So my question is that when we has there been a switch on the data generation side? Because now the AI enables us to use so much of data, but then there's so much of research happening all over, even in the respective organizations. But then is there a long-term thinking about that, okay, now that we can do so many things with AI, then is there at the fundamental level, at the basic level, the way the data is being generated, or even the decisions based off, okay, what experiments do we keep on going? Is there a long-term thinking that, okay, maybe we keep on doing this, then in near future, this might be a database which can be really valuable, because I think that's really important to have that kind of database for the future AI use too. So is there any switch?
Yeah, maybe I can answer this, right? There's definitely a change in the mindset in terms of the data that we are generating, right? Because AI is enabling us to be able to consume this data, right? If we were doing this in old standard statistical methods from the past, there's no way we are going to be able to consume so much data. We are going to just overfit it to the heck, right? So now there is purpose-driven, like what is the data that we need to generate based on what the AI can enables us to do. Right here again, as I mentioned, well, we have to move away from this. Like there has been thousands of years of natural language and the content that has been developed for understanding the foundation models on what the natural language can do and infer from it. But that's not been the same case for biology, right? So we have very limited data, right? We could give all of the species' DNA sequences to a DNA birth model or a foundation model, but it can only see what is the ground state of any DNA sequence, right? You have to be able to look at various complex diseases. What are the DNA changes? What are the transcriptomic changes? What are the phenotypic changes in order to be able to understand and reveal the biology? So I feel like to go to say, okay, we are going to generate all of that data that is necessary for a single foundation model to understand biology is not going to be possible, or maybe it'll take thousands of years, right? Just like what we did for natural language. So as I mentioned, now we are going through a purpose-driven data generation based on the hypothesis that we wanted to solve for. like what I answered for the digital twin, we cannot just start sequencing or generating all of the data to understand, like create a digital twin now. At least to be able to say what are the cell-cell interactions. Yes, that data we can generate, and we are generating those right now in order to, for a model to predict how two cells or particular cells interact and how the pathology changes, right? So that's what we are starting to think as what is the next set of experiments that we have to run to improve these models step by step.
Thank you, Dr. Sowmi. I did check the online questions. There are none. So I think that's good. Thank you so much for your time, Dr. Kevin, Dr. Sowmi, Dr. Pauline, and Dr. Laure. And thank you to the audience for amazing engagement. Appreciate your time. Thank you again.